All right, in the last video, what we were doing is we were talking about the actual cellular response using ADH and aldosterone as an example. Okay, so in this video, what I want to do is look a little bit more closely at the signal transduction. And this is when those signals get from the outside of the cell to the inside of the cell and cause that cellular response. So in the case of the signal transduction, you can see here, here's a receptor embedded in the cell membrane. And when this binds the ligand, you get a series or a cascade of events that's actually going to result in this cellular response. And usually what happens is you get the phosphorylation and the activation of a molecule, and then the dephosphorylation as it relaxes. And then you're just going to pass this signal all the way to the center, to the nucleus of the cell, where you're going to get a response. And with the lipid soluble type of a signal, you don't need that intracellular receptor. It can pass right through the membrane. It binds with, or I should say, you use the intracellular receptor. You don't need the receptor embedded in the membrane. So this can pass freely through the membrane. It can bind with the receptor inside the cell, and then that's usually going to act as some sort of a transcription factor or something that's going to turn on the expression of the gene. You're going to get the messenger RNA and the protein being produced. Okay, so with signal transduction, generally what you have is a multi-step type of a pathway. And these steps here are including, but certainly not limited to, the addition of a phosphate group to a molecule to activate it using a kinase enzyme, or the removal of a phosphate group to often inactivate it using a phosphatase. And then the third kind of way that happens in these things is you get these small molecules or ions, which often act as second messengers to kind of relay that signal. And you could have any combination of these things that are really resulting in this signal transduction pathway. And as we spoke about in an earlier video, what you can see is you've got this single molecule on the outside that gets amplified and leads to this massive response on the inside. So what we were talking about in an earlier video is how like epinephrine leads to the release of a lot of glucose molecules from the glycogen. So it's promoting the glycogen breakdown, okay? So if you remember, the first step in the pathway, of course, is where that signal is getting received by the receptor. And then from there, these relay molecules are going to result in usually a change in the shape of the protein, activation of the protein, something like that. Or you're going to somehow stimulate the cellular response from those pathways, okay? And so we spoke briefly a second ago about kinases and phosphatases. So the phosphorylation and dephosphorylation is one of the really common ways of regulating the activity of the cell and the signal transduction pathway that occurs. So these protein kinases, these are enzymes that are going to add a phosphate group from an ATP molecule and to a protein, and then that's going to activate it. And they're really important in this process just throughout any cells, because at least 2% of our genes are going to be coding for these protein kinases, okay? Now, the other class of molecules are the phosphatases, and these remove the phosphate group from the proteins. And usually this process of dephosphorylation is going to act to kind of shut off the protein activity, or ramp it down in a way so that it's not always on. And so the cell itself might have as many as 100 different types of protein kinases within them to regulate a whole variety of different pathways. And it might not have as many phosphatases. So the kinases are very specific for the pathways that they're activating, and the phosphatases are a little bit more indiscriminate in their ability to shut these pathways off, okay? So if you look here again, this is one of the pictures that we just looked at before. And so here is the activated relay molecule, and that's going to activate this protein kinase. So what that's going to do is that's going to take, it's going to become activated when it accepts a phosphate group from this protein kinase, or onto the inactive protein kinase. And so now it's going to relay or transmit the information to this second step in the pathway. And then from there, you've got a phosphorylase, this thing that's called PP right here, the purple thing. This is going to take the phosphate off of this activated protein and inactivate it. It's going to return it to its inactive form so that it can then, take more information at a later time and so forth. So all of these series of pathways is going to occur until you get the cellular response at the end. And this is the phosphorylation cascade that's in your reading that we talk about in class sometimes, okay? And so you can see how this is the same thing, how you're just getting to the end of it. And the words on this video are the things that we've discussed a little bit in class. So now the second class of things involved in these signal cascades are these second messengers. And these things are not proteins. These are generally small water-soluble types of molecules, ions, things like that, that can very easily move through the cytoplasm. And these participate in the signal pathways that are initiated both by tyrosine kinases and GPCR pathways. And two really common ones are cyclic AMP and calcium, okay? So the cyclic AMP is basically when the ATP molecule gives up its phosphate groups and it's only got one left, there's an enzyme, adenylyl cyclase, which is basically going to kind of make this joined in a way. So it's kind of taking on a cyclic or a repeatable kind of a circular type of a form. And then phosphodiesterase is the enzyme, which is going to convert this back into AMP, and then it'll go through some pathways and get converted back into ATP and so forth. Okay. So cyclic AMP, the bottom line here, the thing you need to know is it's a very common second messenger. You don't need to know much other than that. When you look and see how it is a second messenger, here's a GPCR that's receiving a signal, and then you can see that this adenylyl cyclase is going to make use of all of these cyclic AMP molecules and very rapidly amplify that signal, because each cyclic AMP can activate a protein kinase. So you can see all of these cyclic AMPs are going to activate a whole bunch of protein kinases, which can then go on to activate even more, and so on and so forth throughout that signal cascade to really rapidly ramp up the response. And again, this is the response that we talked about earlier, where you can see the cyclic AMP is going to activate all of those glycogen molecules. And you can see you can activate way more than what we showed if each one of these cyclic AMPs is going to go on to activate a protein kinase. So again, just the whole idea here is using cyclic AMP. It's a second messenger. It's a massive amplification of the response from the outside of the cell to the inside of the cell. And then what happens is you have the phosphodiesterase enzyme that will convert the cyclic AMP into regular AMP, and that's kind of the shutoff switch. So that's how you can ramp down the cellular response. Okay, so you can see that it's regulated by the production of the cyclic AMP and then the breakdown of it. So you can turn on and off these different pathways. Okay, so you can look here. Some of the pathways themselves can be regulated just from the standpoint of inhibiting this adenylyl cyclase. So some of them activate it and get the pathway going. And then another way in which you can regulate it is you can have inhibitory types of pathways. So if you guys remember what we were talking about earlier, in I think it was video one of the earlier videos, when we were talking about cholera and pertussis toxin. The cholera toxin led to a stimulated G protein, which activated the adenylyl cyclase molecule, and it just kept it running on high. Well, in this particular case, you can see how the signaling molecule is going to interact with another action, or another form rather, of the G protein, and its action is going to lead to an inhibitory effect on that adenylyl cyclase. And that's with the pertussis toxin. When you interfere with that, you don't have any way to inhibit it, and therefore it's running on high. So what this is showing you here is some of these cyclic AMP can have an inhibitory effect if it's acting on an inhibitory protein, or it can have a stimulating effect if it's acting on a stimulating G protein. So there's different aspects of how this goes through. So, if you have some questions on it, we can always go over this in class. So just, make sure you're writing them down, and we'll address it further by going back into earlier parts of the video and so forth. All right, so now we mentioned other forms of second messengers, calcium ions, which we talked about a little bit with synaptic signaling, and IP3, which stands for inositol triphosphate. So you just need to know it as IP3. And what happens here is a lot of times some sort of a signal is going to increase the concentration of these molecules inside the cell, and they're going to act as the second messenger. So as we were talking about with the synaptic transmission, generally the calcium concentration is about 10,000 times lower on the outside of the cell. So any sort of activity which is going to open the calcium channels and allow the calcium to rush in is going to increase the calcium ion concentration inside. And then once this calcium ion concentration increases into the cytoplasm of the cell somehow, that's what's going to activate these different cellular pathways that are taking place. Okay. And a lot of times with this calcium release in certain types of cells, you have IP3 and then DAG, which is diacylglycerol. You can see, whoops, hit the wrong one, this diacylglycerol right here. These things are going to be involved in the pathway that's going to lead to the release of the calcium concentration, or the release of the calcium ion, increasing that concentration within the cell. Okay, so calcium ions, again, they can cause muscle contraction, they can cause hormone release, they can cause neurotransmission. And then in plants, what they can do is they can lead to the greening of the plant. So for instance, if you've ever seen the plants that grow the little white things off of them, they're basically in search of light, more or less. And as soon as they find the light, the greening process takes place. So the activation of some phytochromes from the light is going to stimulate the release of calcium ions into the cell, which is going to lead the production of the genes, or the expression of the genes that are going to lead the production of chlorophyll, so that can turn green. So this is something that we will talk about kind of in a later presentation when we get to plants and such. But again, you've got an animal and a plant example here, and how calcium, just more superficially than anything, how calcium is involved in a second messenger that's resulting in a cellular response, either an action potential or the greening of the plant. Okay. So when you talk about the IP3 and the DAG, these are often associated, not always, but often associated with a G protein coupled receptor. So it receives the signal, activates the G protein, and then you can see there's another enzyme in here that's going to activate the diacylglycerol and the IP3, which is going to then again lead to this calcium response. So there's just a little bit more of a simplified version of what we were talking about here. And the important thing is really to know that the calcium and the IP3 and the DAG, these things are all involved as second messengers. The first messenger is the signaling molecule, and then the intercellular response is usually these small molecules or water-soluble ions that are involved in coordinating the other portion of the signaling pathway, which in this case, it leads to the release of the calcium, which can then lead to some sort of a cellular response. With neurotransmission, that's the release of the neurotransmitters. Okay, so now in terms of the response, of course, this is the things that we've been kind of sprinkling in all throughout the way: the expression of the genes, the release of the neurotransmitter, the greening of the plant, et cetera, et cetera. So when we look at these cellular responses, these are just basically ways, the takeaway here is the way in which we turn the genes on and off inside of the cell, okay, from using this signaling molecule. Okay, so there's some ways now with the actual cellular response that we can kind of fine tune it. In other words, we can kind of tweak it to make sure that it's not too powerful of a response, or that we can kind of keep it running at a low level so that it can very quickly be used if we need it, you know, like in mass and so forth, to get the desired cellular response. So the important benefits of using these second messengers is that we can easily amplify the signal, and we can also regulate it. And this is kind of the fine tuning at different points to ensure that we're not wasting energy and we're maximizing the use of the resources within the cell. So basically with this signal cascade, or this signal amplification rather, what we're doing is you have to look at this like a nuclear chain reaction here. So you get a little bit leading to a whole lot of cellular responses. Okay, so that's the whole idea of the amplification. And during the amplification now, there's a lot of different ways in which you can kind of control these different pathways in terms of when you're getting the cellular response out of this. And I'm not really going to go into any of the details that are associated with this figure, but you can see that there's certain parts of the pathway that when they get activated, they can either stimulate the synthesis of molecules that can lead to, say, the storage of excess energy here in the case of these fat molecules. Or if we're going to store this because we have excess energy, then what you can do is you can inhibit other pathways that are involved in breaking down stored molecules that are used for energy. So for instance, just If we want to store the excess energy as fat because we have enough glycogen, then what we can do is we can slow down or stop this glycogen synthesis and store this excess as fat. And at the same time, if we're storing things as fat, we might not want to be breaking other compounds down because we already have enough energy. So the main thing is it's a different regulation point. It's a way for us to kind of conserve resources and maximize the potential of the cell. Okay. So in terms of using all of these relay molecules, what these things are proteins that are basically going to help kind of scaffold or increase the efficiency of this signal transfer from the outside to the inside. So sometimes they might not be able to diffuse quite fast enough so to produce the desired response. So what you might have are some proteins kind of along the way that are going to help kind of coordinate, speed up, or kind of branch out and have multiple effects on the inside of the cell. Okay, so that's what these scaffolding proteins can do. Now, when we get to the end of the response, when we have signal termination, this occurs when the signal kind of reaches its useful life. So in other words, we want to shut this process down. We want to inactivate this. We have enough of a cellular response. We have to have a way to kind of remove the response of the stimuli and then perhaps maybe wait for more at a later time. So when you look at this, some of the ways in which we can have this signal termination is we can either remove the ligand. Okay, so somehow it can get removed from the receptor molecule, or we can modify that receptor in a way so that it no longer accepts the molecule, okay, the signaling molecule, or we can bring the receptor in through endocytosis. We can just bring it into the cell and that will shut down the response because there's nothing there now to receive that signal. Some of the things inside the cell can remove the second messengers. We can pump the calcium ions out or back into the sarcoplasmic reticulum in the case of the muscles and things like that. Or we can have these phosphatases that are going to remove phosphates. Okay, so there's a variety of different ways that we can shut down this signal, and it's important to do that so that we don't have, as you saw earlier on, some of the symptoms associated with the disease. If you have an overactive system, it leads to bad outcomes. So you got to have a way to kind of not only regulate this, but shut it down as well. Okay, so if you have any more questions on any of this stuff that we went over, make sure that you guys write them down. We'll talk about them in class and go over and make sure you guys understand. Okay, so we'll talk to you soon.